Tools of the laboratory. Methods for culturing and microscopic analysis of microorganisms. Methods of culturing microorganisms. The 5 I's. Inoculation, incubation, isolation, inspection, identification. The 5 I's represents 5 basic techniques to manipulate, grow, examine, and characterize microorganisms in the laboratory. First step is inoculation. To grow or culture microorganisms, one introduces a tiny sample into a container of nutrient medium, which provides an environment to which they multiply. This process is called inoculation. Incubation. Once a container of medium has been inoculated with a specimen, it is incubated, which means it is placed in a temperature-controlled chamber, an incubator, to encourage growth. And then we have isolation. Techniques that are based on the concept that an individual bacterial cell is separated from other cells and provided adequate space on a nutrient surface. It will grow into a discrete mound of cells called a colony. Inspection. The colonies on the agar or broth cultures are observed macroscopically, possibly with the aid of staining. Includes such things as looking at a colony morphology, that's the shape and color, and examining the growth inside broth tubes. Some bacterial species grow evenly throughout the fluid, while others tend to grow at the top of the fluid where it meets the air or it aggregates throughout the broth. And then we have identification. The identity of the isolated microbe is determined usually to the level of inspection may be enough to identify some microbes, but additionally technically include biochemical test, immunologic test, and genetic analysis. Physical states of media include liquid media, semi-solid media, and solid media. Liquid media are water-based solutions that do not solidify at temperatures above freezing and that tend to flow freely when the container is tilted. These media, termed broths, milks, or infusions, are made by dissolving various solutes in distilled water. Semi-solid media. At ordinary room temperature, semi-solid media exhibit a cloth-like consistency because they contain an amount of solidifying agent, the agar or gelatin, that thickens them but does not produce a firm surface. Semi-solid media are used to determine the motility of bacteria and to localize a reaction at a specific site. Solid media provide a firm surface on which cells can form discrete colonies and are useful for isolating and culturing bacteria and fungi. Liquefiable solid media, sometimes called reversible solid media, contain a solidifying agent that changes their physical properties in response to room temperature. Here is a visual example of liquid media, semi-solid media, and solid media. The first column is the liquid. You can see as you tilt it, it moves. The semi-solid is the second column. Third column is the solid reversible to liquid. Selective and differential media. Selective media are very important in primary isolation of a specific type of microorganisms from samples containing dozens of different species. Samples are feces, saliva, skin, water, and soil. Differential media allow multiple types of microorganisms to grow, but are designed to display visible differences among their colonies. Differentiation shows up as variations in colony size or color, in media color changes, or in the formation of gas bubbles or precipitates. Example: when microbe X metabolizes a certain substance in the medium not used by organism Y, then X will cause a visible change in the color of the colony or the medium, and Y will not. Defined and complex media. Complex media are rich in minerals and nutrients and are often used for growing a wide variety of microorganisms, including fastidious ones. Defined media, on the other hand, consist of ingredients whose chemical formulas are known. And here we have the metric system. The giga is a billion, mega is a million, kilo is 1,000, the meter is a meter, centa is 100th, milli is 1,000th, micro is 1,000,000th, nano is 1,000,000,000th. And here is a view of the macroscopic view, 1 millimeter, the range of a human eye. It shows the louse and the reproductive structure of bread mold, all the way down to from microscopic view down to 0.1 nanometers, which is a hydrogen atom or amino acid, small molecule. That's a good example. Earliest microscope. The first microscopes were simple, meaning they contained just a single magnifying lens and a few working parts. Examples of this type of microscope are a magnifying glass and a hand lens. Good elements of microscopy. To be the most effective, a microscope should provide 3 properties. Those 3 properties are magnification. In most microscopes, results from a complex interaction between visible light waves and the curvature of the lens. And resolution, distinguishing magnified objects clearly. And contrast, the third quality of a well-magnified image is its surrounding. And here is a picture of a microscope, some things that you would need to know to use that, to look under, to see the plates and stuff. Light versus electron. Electron microscopes differ from light microscopes in that they produce an image of a specimen by using a beam of electrons rather than a beam of light. Electrons have a much shorter wavelength than visible light, and this allows electron microscopes to produce a higher resolution images than standard light microscopes. And in this slide we have 3 types of stains. Column A is simple stains. Crystal violet stain of E. coli, Listeria coli. And then we have the methane blue stain of Trichomonas. And then we have the colochlor stain of Candidaceae, Cannabaceans. The column B is differential stains. This is gram stain, purple cells, gram positive, red cells are gram negative. And then acid fast stain, red cells are acid fast, blue cells are non-acid fast. And then the endospore stain showing endospores in red and vegetative cells in blue. And then the third column we have special stains, India ink capsule stain of Capsoxis. And then the last one is the flagellum stain of Proteus vulgaris. A basic stain was used to build up the flagella. So there is a example of the stain that you would see under a microscope.